for what we're doing today is I'm going to kind of review some idea. Our pathways that we're going to focus on are involved in glucose oxidation and energy generation. At the end, we get a little bit of lipid metabolism and a little bit of amino acid metabolism. But most of the metabolism we talk about is energy metabolism in terms of the overall oxidation of glucose. And so this third little section of this introduction I'll talk about is a review of the concept of oxidation. Chemists can talk about oxidation in several different ways. They generally talk about it as the removal of an electron from a system, right? Like you talk about the oxidation of iron, say going from Fe2 plus to Fe3 plus. We removed an electron. That's what oxidation is, okay? But we also talk in terms of carbon metabolism. We talk about oxidizing it, going from an alcohol to an aldehyde as an oxidation process, going from an aldehyde to a carboxylic acid as an oxidation process. But we don't necessarily see the removal of the electrons quite as easy. So what's going on in there? And it basically arises from the fact is that we consider in a carbon-oxygen bond that the electrons are shared so unevenly that when you have electrons in a carbon-oxygen bond, you might as well consider that the oxygen has taken the electrons away. So if you have a carbon, actually let's go ahead and give an example. So if we have, I'm going to start with ethane. And we compare to going to an alcohol. That in this system, let's count the electrons that are in these bonds here. So we've got 1, 2, 3, 4, 5, 6, 7. So 14 electrons, right? 14 electrons. Now, if we go to the alcohol, what we're saying is that this pair of electrons, we're not going to really kind of count that as being associated with the carbon, right? That in fact that this pair of electrons kind of is so dominated by the oxygen that when we count, we're going to count 1. Here's 1, 2, 3, 4, 5, 6. So this basically have 12 electrons. So the carbon is essentially lost 2 electrons because the oxygen is basically hogging it when the carbon-oxygen bonds.
And this is due to just the electronegativity
of oxygen. Exactly. Yeah, exactly. And as a matter of fact, this alcohol has the same oxidation state as ethene. One, 2, 3, 4, 5, 6. Both of those have sort of 12 electrons that are associated with it. So we say that the alcohol has basically the same oxidation state as ethene. And we could keep going. I'm not going to count all the electrons, but just a little reminder that if we can further oxidize it, taking away another 2 electrons to make acetaldehyde here. Actually, I guess I could go ahead. 1, 2, 3, 4, 5. So this is 10 electrons here. It would actually be the same oxidation state as ethine or acetylene. And we could keep oxidizing it, taking away another 2 electrons. To make the carboxylic acid, acetic acid. This only has 1, 2, 3, 4, 8 electrons. It's not that there aren't electrons there, but they're just associated with bonds to oxygen.
Professor?
Yes.
Isn't ethene supposed to have an H right there to the bottom left?
Thank you. So essentially, what the when we oxidize something, we're going from here to this direction, we're exchanging carbon-hydrogen bonds or carbon-carbon bonds for carbon-oxygen bonds. So let's look at the overall oxidation of glucose. So what we have is, this is the overall oxidation of glucose. Takes C6H12. This is the thing that you learned. Plus 6O2 goes to 6CO2 plus 6 water. Right. That's the overall oxidation of overall oxidation of glucose. Now let's look at really what's going on here. I'm going to draw the structure of glucose. I'm going to draw it in a way we can actually see all the hydrogens. And we're going to end up making six carbon dioxides. And if you think about, okay, let's count up the number of, first of all, the number of carbon-carbon bonds. 1, 2, 3, 4, 5. There are 5. The number of carbon-hydrogen bonds. 1, 2, 3, 4, 5, 6. Oh, I thought there were 7. Did I miscount? 1, 2, 3, 4, 5, 6. No, I got 6. Number of carbon-oxygen bonds. 1, 2, 3. Actually, I better count this one. 1, 2, 3, 4, 5, 6, 7.
You're missing a hydrogen on carbon 5.
That's it. I knew there was, I thought there was 7 here. All right, now over here, what we have is what, 24 CO bonds, and none of those. And then this is energetically more stable than this is. So with the process of oxidation, the complete oxidation of glucose is basically to convert carbon-carbon bonds to carbon-hydrogen bonds and carbon-oxygen bonds. All right? And because the carbon-oxygen bonds are more stable, it's more energetically favorable. And so we can couple this process to the synthesis of ATP. Anybody remember how many ATP this gets coupled to?
Like 6 or something.
And it's okay that it's like, because it kind of depends on how you count. We'll get to that in a little bit. But I'm going to say more or less 34 ADP plus PI to 34 ATP. When we get to it, we'll see that it's not actually a stoichiometric process. That there are some different things happen to make that number vary. One reason I wanted to review this is because we'll be getting to some reactions to say, okay, this is an oxidation reaction. So you can go back and look and see, okay, well, how did the number of carbon-oxygen bonds change in this to be able to see that? And again, generally when there are carbon-oxygen bonds that, when there are carbon-oxygen, when you have an oxidation reaction, it's going to be energetically favorable and usually couple that to synthesis of ATP, or the synthesis of another molecule can easily be used to synthesize ATP.
Professor, what's in front of the 34 ADP plus.
Oh, and more or less a squiggle.
Oh, okay.
A squiggle. All right. Now what I'm going to do is I'm going to give an overview, like one way of doing it in this class is I could dive right into glycolysis. That's the first part of the oxidation of glucose. But rather than do that, I want to paint for you the whole picture of where we're going in, and then come back and we'll talk about the specifics of the different pathways. So I'm going to give an overview that includes glycolysis, pyruvate dehydrogenase in the citric acid cycle, and the electron transport chain to see basically how this whole process occurs. And then we're going to dig into the details. So I'm going to draw a big picture of a cell. I'm going to spread the cell over like a board and a half here. There's our cell. Generic, we'll call this a liver cell or something. Here's a generic cell. Then the other thing I'm going to do, and I'm going to draw in a way that is totally unrealistic. Don't just show this to like any cell biologist or anything like that. But I'm going to draw a mitochondria. Here's a mitochondria. So over here we have the cytosol. And over here we have the mitochondria. Okay, so say this is a liver cell, and we've just picked up some glucose, some dietary glucose from your bloodstream. So we have our glucose here. The first part of this is going to be the process of glycolysis. What glycolysis is going to do is it's going to take the glucose molecule, split it in half, and then also oxidize it just a little bit. So what we're going to do is we're going to come in here, we'll go through, so there's 10 reactions of glycolysis, and we're going to generate 2 pyruvate molecules. Now, in the process, if you would count up all the electrons associated with carbon in this system, again, not counting the ones that have bonds to oxygen here, like this, and then you can count it up to here, you would finally, there's actually 4 less here than there are here. Those electrons that were taken away end up going to a high energy electron carrier known as NADH. Now, I'm going to wait to talk about the structure of NADH and how that works until we actually get to that reaction in glycolysis. But I just will put this is a high energy electron carrier. to? Where's it carrying those electrons to? I'll get to that. The other thing that happens in this process, because we've done a little bit of oxidation, so we generate at a net reaction, we'll get into that more, is we take 2 ADP plus PI and make 2 ATP. So we couple the oxidation of the pyruvate to the synthesis of ATP. We've taken some of the electrons that happened during that oxidation, and we're now carrying them with this molecule NADH. All right, now what happens? Well, again, what we're trying to do is we're getting to 6CO2. Now the reaction comes this way. That pyruvate comes in here. And here's our pyruvate. Pyruvate comes into the mitochondrion. And the first step, the reaction catalyzed by an enzyme complex known as pyruvate dehydrogenase, we bring in a molecule of coenzyme A. I'm just going to abbreviate it. Again, when we get to that, we'll get into all the lovely details about that. HS-CoA, coenzyme A. And we're going to do our first complete oxidation of a carbon. The carbon is going to come off as CO2. Now, all of this, per glucose molecule, all of this happens twice. I'm just drawing one pyruvate, but remember, glucose generates 2 pyruvate, but I'm not going to put 2s on everything. So out of the 6 CO2 that we're going to generate, we've generated now 2 of them. We're a third of the way there in terms of generating CO2. This oxidation is coupled, those high energy electrons, again to this carrier, NAD. This high energy now those electrons, like this, oh, then what's the product? The product here is acetyl-CoA, which I'll draw like this. This is acetyl-CoA. And this process right here is known as pyruvate dehydrogenase.
Wait, which part is the pyruvate dehydrogenase?
So that's the enzyme that catalyzes this whole process. It's actually an enzyme complex.
So the HS-CoA is the pyruvate dehydrogenase?
No, this is coenzyme A. Again, and we'll get to the nitty-gritty details of this. The enzyme that catalyzes this whole reaction is pyruvate dehydrogenase.
What's this whole process called?
What's what?
This whole process called?
So what I'm like this is going to be pyruvate dehydrogenase / the citric acid cycle. So I just started the pyruvate dehydrogenase, and I'm just getting ready to show you what the citric acid cycle does. This whole process, I didn't write this whole process. This is glycolysis here.
So, Professor, you're ending up with 2 of those acetyl-CoAs?
Yes, per glucose molecule, exactly. Now we go through a series of 7 different reactions. And I'm not going to go through all the details here. known as the citric acid cycle. And it's basically going to finish the oxidation job. So, two of them are going to generate 2 carbon dioxides. And we're also going to generate some more of these high energy, these are oxidative reactions. We're going to generate some high energy electrons carriers. There's another one that generates a different high energy electron carrier known as FADH2. FADH2. So this is another high energy electron carrier. And there is a one of these enzymes also, I'm not getting the order exactly right, but another one will take GDP plus PI, so it's an ATP equivalent, and make GTP. So it's just like ATP except the base is different, guanine rather than adenine.
You said that was what, guanine on the GTP?
The G, yeah, guanine. People kind of ignore that difference. Like you don't, when people talk about this, this is really like 33 ATP plus 1 GTP, but people just kind of ignore that and say, well, energetically it's the same thing as an ATP, so we'll just call it an ATP. Okay, so now we've completed this process. We've taken our glucose molecule between pyruvate dehydrogenase and the citric acid cycle. We have totally oxidized that that molecule to that glucose molecule to carbon dioxide, right? We can breathe out, right? That's why we breathe out, right? Is all of this is the CO2 coming from there, there, and there. But how much ATP have we made? Well, we made 2 ATP here, and since this happens twice per glucose molecule, we've made 2 GTPs here. Nothing like the 30 or 34. That's because most of the energy that has been coupled to the oxidation of this is tied up in our high energy electron carriers, NADH and FADH2. So the next process takes the NADH that is coming from there, that is coming from here, coming from here, and even the FADH2 coming from here. So I'm going to call this NADH, FADH2. These are our high energy electron carriers. And those high energy electron carriers, in a process that's really cool, and we'll get there. This is a disservice. These arrows are a disservice to the coolness of the process. Those high energy electrons are ultimately go to an oxygen to make water. And we go back to our NAD plus FAD, our oxidized version of these high energy. So those electrons that are in there get coupled to water, and in the process, we couple that in a process called oxidative phosphorylation to 30 ATP. So this whole process down here is electron transport. We're transporting the electrons from NADH and FADH2 to oxygen to make water. And this and oxidative phosphorylation. That's what this is here, oxidative phosphorylation. We're coupling the synthesis of ATP to the reduction of molecular oxygen to form water, or to the oxidation of these high energy electron carriers. That's probably a better way of describing it. The oxidation of those high energy electron carriers. We're coupling the synthesis. All right, so this is the process. We can take a deep breath, because this is where we use oxygen, right? So this is why we breathe. I mean, this is why I love the biochemistry and how it relates to physiology. This is why we breathe in, right? This is why we breathe in. This is why these CO2s, that's why we breathe out, right? This is why we have blood, right? Because we got to carry that oxygen and carry that carbon dioxide away. It's why we need to have heart, so that we can pump that stuff around.
So,
You know, what's that?
Relating to this specifically, if sugar is what fuels this process, is this the primary reason why I say something like low blood sugar is bad, because it can't do that without the sugar? Like,
Yes, exactly. And particularly the brain relies almost exclusively on glucose as an energy source. Other cells can use fats as an energy source as well, and we'll get to that process. That's called beta oxidation. But the brain doesn't use fats as an energy source. It can only use glucose. So what happens is if your blood glucose goes down, right, then you're not generating ATP to generate that sodium potassium pump we talked about earlier, to generate the electrochemical gradient in your brain, right? I mean, it is pretty amazing to think about the brain. So say we can't breathe, right? How long does it take before this brain stops functioning, right? Well, we go without that for what? Maybe 7 minutes, something like that. We go like this and we're kaput, like this. It's kind of amazing the importance of this whole process to our ability to continue to reproduce, I guess, from a biological standpoint.
And just to be clear, you were saying glucose is like the real sugar, like that's the one we want, none of this fructose, lactose stuff?
Yes. So glucose is the main. Now you can get, you can actually, fructose will work. I don't know if the brain uses fructose very much. I know other tissues does use glucose, but you use fructose. But glucose is the main one. All right. Now, so what we're going to be doing is on, let's see, on I think it's on Thursday, we're going to be going over glycolysis and pyruvate dehydrogenases, and probably getting the citric acid cycle, and then next we're going to be going into the oxidative, the electron transport and oxidative phosphorylation. But there's something else I'll kind of go through, is give you a little bit of an idea of sort of the other pathways that we're going to be talking about that get mapped on top of this. One is there are certain cells that don't do this oxidative stuff, or do it very little, okay? And think of that as your fast twitch muscle fibers, right? It's sort of the white meat muscle fibers. Those are the ones that are important, like for sprinting, and basically they, you use ATP so fast that you can't supply enough oxygen fast enough to drive that process. That's right. So you basically, this is the 100-meter dash sprinters, right? They're just going like crazy. You don't have enough oxygen to actually supply that ATP cleavage fast enough through this process. So you strictly derive your ATP just from glycolysis. You can do this fast enough, right? You don't need oxygen. You can do this fast enough that you will actually can generate enough ATP until your glycogen runs out, and then you're kind of done. So then the question becomes, what happens to the pyruvate if it's not getting consumed by the in the mitochondria in oxidative phosphorylation? Well, then this ends up getting converted to lactate. Lactate, or actually, I could draw how it is physiologically with a negative charge. Lactate, or if I put the proton in there, it'd be a lactic acid. Okay, so that's lactate. Okay, and actually, what that ends up generating, that goes back the other way and takes NADH and makes our NAD plus, and then that can actually feed back up in here. So it basically makes a closed loop, and you end up accumulating lactate. Now, there's more detail about that. We'll get there when we get in there, but it's kind of fun from an exercise physiology. So that happens. So here's some other things that we'll talk about briefly, interact with this. There's an intermediate in here that gets branched off to form glycogen. So we have glycogen synthesis and degradation. All right, there's one other thing that we're going to do is we're going to, that we'll talk about to a certain extent, is what if we don't have enough dietary glucose, right? What if all we did was we eat the sausage for breakfast, we didn't have any bread or anything, no carbohydrate, we don't have any dietary glucose. The brain is still going to need glucose, all right, but we just ate fats and amino acids, right, in our diet. Basically, we can draw from the amino acid cycle. Amino acids can enter the citric acid cycle. We can draw from those, and we can end up running this process in reverse. And that process is called gluconeogenesis. So we'll be talking about gluconeogenesis as well. All right, so that's the end of my introduction to metabolism. I do think I want to take, because with losing that day, I don't want to. I do want to at least. I'm going to get started on glycolysis. So we're going to first start on glycolysis, and I'm just going to kind of give. It's more overview material. I'm going to kind of give overview material on glycolysis, and then on Thursday we're going to get into the individual reactions, and I can jump right into the individual reactions. All right, so this is a new outline, glycolysis. We'll start with an overview. And in the overview, we're talking about there's two stages. There is the energy investment phase, and there'll be the energy recovery phase. In the energy investment phase, we're going to take our glucose molecule here. And we're going to add phosphate to it, and we are going to add phosphate to it, and we're going to split it in half, so that we end up with 2 glyceraldehyde 3-phosphate molecules. And I'm going to I'm going to draw them like this, because that's the way we'll draw them. Do this. So we're going to end up splitting this molecule 1, 2, 3. We're going to split it here. 1, 2, 3. We're going to split this bond right there. And except I forgot to put the phosphates on here. OP, and I'm going to put the P with a circle. That's my abbreviation for phosphates. In order to do that, in order to make these phosphate esters, that's going to take some energy. So we couple that to ATP, so we basically have 2 ATPs plus, and then we end up getting 2 ADPs. Might say, hold on, what's going on? This is we're supposed to get ATPs, not use it up, right? But in this case, in this first phase, it's an energy investment phase, we actually use some ATP. All right, so the next part, the next phase is going to be the energy recovery phase. And that's where we're going to take our 2 glyceraldehyde 3-phosphates We're going to take 2 of those, again, go through a series of reactions, so we'll get into detail here, and we're going to make 2 pyruvate molecules. So you might notice the pyruvate molecules don't have any phosphate attached. So we're going to end up using those phosphates to ADP to make 2 ATP. We're going to remake our ATPs. The other thing is we're going to use the energy to make another 2 ADP plus PI to ATP. So that our overall yield in this process is 4 ATP. Two from using this phosphate, and then 2 just using the energy to be able to make the ATP. Then the other thing that happens in here is there's an oxidation involved, and we make and this all happens twice, we make 2 NADH, our high energy electron carrier, that ultimately will go to the electron transport chain. All right, so on Thursday, what I'm gonna do is this whole process between the energy investment phase and the energy recovery phase, there are 10 reactions. We're gonna walk through reaction by reaction. We'll use this as an opportunity to kind of look at enzyme mechanism a little bit, remind ourselves of these different reaction types, the hydrolases and all of that stuff. But then what I'm also hoping to do is to try to give you a little bit idea of sort of the chemical rationale for why the pathway exists the way it does. So we're not just like rote memorizing reactions, but we kind of see the rationale of the chemistry that's actually involved. All right, so we'll go ahead and stop there for today. Got plenty of studying to do for the exam tonight. Remember, 7